Welcome to Study Loop.
Hi everyone, my name is Eric and let's go over problem 36. In this question, we are going to be focusing on alternative splicing. So in order to answer this question, first we have to define what alternative splicing is. So first, let's just look at the word itself, alternative splicing. Now, alternative means different, so it's like a different method, different from the usual. And splicing just means to cut up. Essentially, splicing is to split or cut. And so, what alternative splicing is a method to create new proteins. So, from the central dogma, we know that the DNA follows a path through transcription to make mRNA, and then through translation, we can make protein. Now, DNA cannot easily be altered, so how can we get different forms of protein? Well, this is where alternative splicing comes in, where we can cut up the mRNA in different combinations to give us different proteins. So, for example, let's say that here is our mRNA right here. Now, this mRNA is going to be sectioned up into 1, 2, 3, 4, and let's say five different exons, right? Now remember that exons are the coding sections of an mRNA. They actually code for the protein, right? And there are combinations of exons and introns. Alternative splicing is a method where the protein that is responsible for it will come in and splice. It will cut up this mRNA and assemble it in a different order. So let's say that one order could be 1, 2, and 5. And let's say a different order could be 2, 3, and 4. Now, we know that these are two different mRNAs, as first off, they're shorter, and second, they have different exons, right? And each exon combination will give us a different protein. So let's say that this mRNA will be translated and will give us a green protein. Whereas if this mRNA was translated, it would give us a blue protein, right? And this is the method of alternative splicing, where we can take our mRNA, put it through different combinations to give us new types of protein. And so after looking through the answer choices, we can tell that it leads to the synthesis of something. And that something are proteins. And what are proteins made up of? Basically, these are polypeptides. So the answer to the question is A, where we are going to be using alternative splicing to make several different types of polypeptides from a single gene, right? This would be from the process of transcription all the way to translation to get our various proteins. So I hope this helps explain a little bit about what alternative splicing is and how we can use it to make new proteins. Thanks for watching, and I'll see you next time.